Hello, and welcome to this podcast for cell biology. In this podcast, we're going to spend a lot of time talking about G protein coupled receptors, which we will abbreviate as Gpcr. Throughout this podcast, I want to first introduce you to what these G protein coupled receptors are, an intro to that, and the parts of it. Then I want to spend just a little bit of time talking about second messengers. And then finally, I want to close with a specific G protein coupled receptor that makes a specific second messenger. And so we'll spend some time talking about cyclic cAMP, that's the second messenger, and its linked G protein. But let's go ahead and get started with our introduction. G protein coupled receptors. This is an incredibly important family of proteins. So I want to begin by talking about the Gpcr family. All the proteins that function as G protein coupled receptors. And it's a big one. It's the largest family of receptors. Humans have about 700 Gpcrs. Mice have more than 700. In fact, of all the ones mice have, 1,000 of them are involved in the sense of smell, which makes sense. Being able to smell various odorants is essential for the mouse to survive. We don't need quite that many. Now, I wish my kids had about that many. That way they would be able to tell me why their rooms smell the way they smell, but we'll just stick with what they were born with. They can accept a variety of signaling molecules, almost any kind that we've talked about before, but certainly proteins, peptides, fatty acid, and amino acid derivatives, and hormones. Because they're involved in so many different pathways in a cell, they are targets for drug development. This is highlighted by the observation that 1/3 of all drugs used today, legal drugs used today, act through a Gpcr. Finally, I'm going to say all Gpcrs have a similar structure, which I'll draw on the next slide. Okay, so let me draw our ever so famous membrane here. And here's where I'm going to draw the G protein coupled receptor. We have one end of the polypeptide here in the extracellular space, and the other is on the cytosol side. Now it's a G protein coupled receptor, so we should probably introduce the G protein. And first, let's define what a G protein is. It's a membrane bound GTP binding protein. It's involved in intracellular signaling. And lastly, a G protein contains 3 subunits. So let's draw it right here. The first subunit here is the alpha subunit. And you can see that it's attached to the membrane by a lipid tail. So here's the lipid tail right here. It's bound to the other 2 subunits. And so the other 2 subunits are called beta and gamma. The gamma subunit is also attached to the membrane using a lipid tail, shown here. And the beta subunit is not attached to the membrane, but it is covalently bound to the gamma subunit. As long as these 3 subunits are bound to each other, as shown here, though it's not abundantly clear, I'm trying to imply that there is this interaction between all 3 subunits. So this G protein as drawn is inactive, just as the Gpcr. So how are we going to activate it? Well, let's draw that down here below, so I need to erase our definition here. So let's redraw our membrane, and what activates the Gpcr will be this signal here. And so let's draw what the Gpcr will look like once it's activated. So we'll have the signal bind here, and I draw the Gpcr slightly changed, because when it binds, it does cause one of our favorite things, a conformational change. This conformational change will allow the alpha subunit of the G protein to interact with the GPCR. And I should have mentioned, pretty big fail on my part here, that the alpha subunit in the inactive form is bound to GDP. And remember, when G proteins are bound to GDP, that's a signal that it's off. So that's why it's inactive. And so it will, of course, bring along our gamma subunit here and the beta subunit because they're attached to each other. Upon binding to the GPCR, the GDP is lost. Then what will happen is GTP will come into this active site. That does a couple very important things. It will now make the alpha subunit active, and sometimes when we talk about an active subunit, we'll call it GS. And when we talk about an inhibited or inactive subunit, we'll call it GI. This binding of GTP does something else. It releases the gamma subunit, but it's still attached to the beta subunit. Now when that happens, the beta and the gamma subunits are now active as well. Now we'll talk about this in greater detail, but what each of these will do, they will bind to separate targeting proteins in the membrane, and they will activate these membrane proteins, which will then either produce or remove second messengers. And we'll talk about second messengers in just a moment and what they are, but we've never defined what a first messenger is, and let me go ahead and do that now. The first messenger is this original signal that comes along here and starts the whole process. Often we talk a lot about second messengers, but we don't often use the term first messenger. But since I said second messenger, you might have been wondering what happened to the first. So that's where the first is. Okay, so now let's spend some time talking about second messengers. Okay, so let's begin with a brief definition here. These are small intracellular signaling molecules that are generated or released in response to an extracellular signal. Just like we had on the last slide, we showed how the G protein, once activated, would then activate a target protein, which would then help generate or remove these small intracellular signaling molecules. Some examples include cyclic AMP, cyclic GMP, inositol, triphosphate, calcium, and diacylglycerol, which we often just refer to as DAG. So in a very general sense, this is what happens. We'll have our G protein here, which is bound with GTP, so it's active. It will interact with this target protein. And so when it interacts with it, this target protein, this enzyme, it's an enzyme, is now active. And being active, it can convert a substrate into a product. And in this case, the product will be a second messenger. This second messenger now acts as an intracellular signaling protein or molecule. Okay, now that we are done talking about the basic background of G protein coupled receptors, as well as some examples of second messengers, let's now move on to a specific example looking at how cyclic AMP is produced by a G protein coupled receptor, and then what that cyclic AMP does to activate downstream responses. All right, let's get started. But let's begin with cyclic AMP. So before we talk about the role that G proteins have in making cyclic AMP, I want to draw out the pathway here of making cyclic AMP. So cyclic AMP is produced from ATP, and then to inactivate cyclic AMP, it's converted into AMP. As I've mentioned before, the enzyme that converts ATP to cyclic AMP is called adenyl cyclase, which is an enzyme activated by a G protein. And when this happens, we have a very quick rise in cyclic AMP. And we need that quick rise because cyclic AMP is continuously converted to AMP by the enzyme called phosphodiesterase. This enzyme is important because it terminates any signaling that cyclic AMP is having. So any pathway that cyclic AMP is activating is immediately or can immediately be turned off by this enzyme. So which pathways can cyclic AMP regulate? Okay, so now let's continue our discussion of cyclic AMP, but now let's look at the G protein that's linked to it and how we're going to make cyclic AMP. So let's draw our membrane here. We'll draw our GPCR here, and we'll draw it bound with the signaling molecule adrenaline. Now when that happens, remember, that will activate the alpha subunit and the beta and gamma subunits of the G protein. For simplicity, I'm only going to draw the alpha subunit because it's the one that's involved with cyclic AMP production. And so remember, it's going to be bound with GTP since it's active now. And being active, it will interact with its target enzyme. And its target enzyme is adenylocyclase. Adenylocyclase will take ATP and produce cyclic AMP. Now, what will cyclic AMP do? Well, on the previous whiteboard, remember we mentioned that cyclic AMP could target fatty acid breakdown, it could target glycogen breakdown, it can also activate gene transcription, and that's what we're going to talk about here. But it doesn't directly activate gene transcription, but rather what it will do is activate an enzyme called pKa for protein kinase A. And so I'm going to put a little asterisk by it because now it's active. And we can draw that here on the bar here. So it activates pKa. And on the next board, I'll talk more about what that means. Now this activated pKa can now enter the nucleus. So here's our nucleus. It will enter the nucleus, and in the nucleus, it will take an inactive transcription regulator. So I'm going to put TXNREG for transcription regulator, and it will convert it into an active transcription regulator. This active transcription regulator is now able to bind to a promoter and activate transcription from that promoter to produce this gene product, this protein. So let's review this real quick. Adrenaline binds to the GPCR. It activates the G protein. The alpha subunit here is now active. It will in turn activate adenylocyclase. Adenylocyclase will convert ATP to cyclic AMP. Cyclic AMP then will activate pKa. pKa now activated will enter the nucleus. The activated pKa in the nucleus can do several things, but in this example, it will activate an inactive transcription factor into an active transcription factor. This active transcription factor can now bind to the promoter of specific genes and activate transcription and ultimately protein production. And now that that protein is made, it will have a specific response. Maybe that response will be involved in the increase of the heart rate or various other functions. Cyclic AMP targets many different transcription factors that have a variety of functions. Okay, that's all I have for this podcast on the specific G protein coupled receptor that is linked to adenylocyclase and how that produces cyclic AMP. If you have any questions over this material, please make sure you come see me. If not, I'll see you in class. Bye.